Hello and welcome to the online presentation of my poster at ISAPP 2019 in Antwerp. My name is Ludwig Lundqvist and I'm an industrial PhD student at BioGaia, performing my research at the Swedish University of Agricultural Sciences in Uppsala in Sweden. I will be following this agenda or outline, whereas the time point indicated in the circles on the left-hand side represent at what time point in the presentation that particular part is initiated. You can then fast forward to that given time point should you find a certain part extra interesting. On the right-hand side you can see the poster in its full, which you will also find in the end of this presentation. Lactobacillus reuteri is the second most studied probiotic bacteria. That means that to date, there are 203 clinical studies that have been conducted on BioGaia's human Lactobacillus reuteri strains, whereas over 17,000 individuals have taken part in these studies. Live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. That is the global definition of probiotics, and Lactobacillus reuteri certainly confers beneficial effects on the host. In fact, Lactobacillus reuteri have proven to ameliorate a variety of disorders, including infant colic, acute gastroenteritis, childhood functional abdominal pain, along with functional constipation, among others. Along with the growing evidence of microbiota as an important mediator of well-being, we're investigating what mechanisms and key components are crucial for Lactobacillus reuteri's beneficial bioactive properties. My PhD project is part of a larger initiative in collaboration with Lund University, also in Sweden. We are 3 PhD students with different focus areas, namely fermentation, freeze-drying and bioactivity, the latter being my project. So, it is known that adjusting production parameters will affect the storage stability and viability. What is not known is how these production parameters can impact the bioactivity. How can we alter the means of producing the bacteria and thus improve their function or their bioactivity? When referring to bioactivity, I mean the tolerance to physiologically relevant stressors, such as tolerance towards low gastric pH and bile, which in turn indicates whether they will survive the passage of the gastrointestinal tract. But when I say bioactivity, I also mean the host microbe interactions, or more precisely, the protective effects conferred to the host, which is something that we mimic with various cell line models. Another aspect to consider in this context is the question of a potential correlation between increased bioactivity and increased viability and storage stability. Do we somehow affect the viability and storage stability when adjusting production parameters? Probably, but can we improve them along with the bioactivity? Does that somehow correlate? These are questions that we hope to gain insight in the upcoming years. Even though much of which I've said kind of naturally alludes into the aims, I will shortly explain them. The aims are to investigate differently produced batches of Lactobacillus reuteri DSM 17938 in preclinical models. These preclinical models that I'm referring to is, as mentioned, the bile and acid tolerance assays, as well as the host microbe interactions. When I say differently produced batches in this context, it refers to alterations that we made in the production, which I will get back to on the next slide. Furthermore, we will also investigate the interaction mechanism between differently produced Lactobacillus reuteri DSM 17938 and or its bioactive metabolites with various receptors, such as the pain receptor TRPV1. We further also intend to quantitate the effect of potentially bioactive molecules produced in response to these varying growth conditions. I've put quite a lot of focus into the methods section in this poster, and this illustration can look complicated at first glance, but I hope you'll find it well explained in the coming section. So, when producing probiotic bacteria such as Lactobacillus reuteri, you have a core workflow, which is illustrated in the upper part of this slide. It starts with a fermentation, which is one of the 3 points where we can alter the means of production. To exemplify what I mean, we can use different substrates for cultivation, such as in MRS broth or in another media. We can add or withdraw components in this part, which may or may not affect the bioactivity of the product. Following fermentation, we have formulation, which is the step where we suspend our pelletized bacteria in a protective matrix. The purpose of the formulation is to protect against the severe stress and dehydration that comes with freeze-drying. This is often done with a matrix containing some percentage of sugar, such as sucrose. However, this is also a possible step of modification, whereas the formulation can be altered in many ways, not only experimenting with the sugar content, but with other molecules as well. As I said in the background slides, most probiotic bacteria come in freeze-dried form. And that is the final step before we start to evaluate the bioactivity, namely freeze-drying. In the freeze-drying step, we can use different programs, using for instance a more conservative program, making sure our samples don't collapse, or trying with a faster, less conservative approach, preferably with an equal or even better survival rate than the conservative one. It is known that the freeze-drying step is crucial when assessing survival. However, whether it affects the probiotic activity remains to be elucidated. The 3 aforementioned steps lead to the magnifying glass and the box where our primary focus lies, namely bioactivity. I will shortly repeat what I mean when I say bioactivity. I mean the tolerance to physiologically relevant stressors, such as tolerance to low gastric pH and bile, which indicates whether they will survive the passage of the gastrointestinal tract. But I also mean the host microbe interactions, or more precisely the protective effects conferred to the host cells, which is what we mimic with our cell line models. While we assess bioactivity, we also assess the classic parameters involved in process optimization, hence the green feedback arrow to the pipeline. This includes mainly viability and storage stability, which is what probiotics is optimized towards today. However, with this project, we strive to enable not only optimization towards that, but optimization towards bioactivity. That is key in this project. That is what we want to accomplish, to increase the probiotic activity by adjusting certain parameters. Now, to the lower part of this picture, where I will dig deeper into what we actually mean by bioactivity and explain the cell models, and somewhat merge that with the continued work after isolating a parameter that impacts bioactivity. Exploring bioactivity, or finding the components responsible for the bioactivity, here named the probiotic effector molecules, that is our aim. But before going into this, I will explain our cell models more carefully. First off, the 2 adherent cell lines IPEC-J2 and CACO2, which are intestinal permeability cell lines derived from pig and human respectively. In experiments with these 2 cell lines, we seed them onto a filter insert, whereas we allow them to differentiate and form a single cell layer. Following the differentiation, we treat them with our Lactobacillus reuteri and then induce leakage by disrupting the single cell layer with a physiologically relevant pathogen, such as enterotoxic E. coli. As we add the E. coli, we also add a fluorescent molecule called Fitch-dextran. During the total time of stimulation, which is 6 hours, we measure the ionic flux between the filter and the cells and the underlying media. This is called measuring tear, short for transepithelial electrical resistance. After the 6 hours, we measure how much of the fluorescent molecule that has leaked over into the basolateral layer of media. Lactobacillus reuteri have proven effective in protecting against this kind of leakage, which is a property of Lactobacillus reuteri that we want to enhance. This method as a whole simulates leaky gut syndrome. Our second model is in a Jurkat T cell. This model is in a suspension rather than being adherent, and here we use capsaicin to induce a pain sensation in a pain receptor expressed by these T cells, called the TRPV1 receptor, which is short for Transient Receptor Potential Valenoid 1. We investigate whether our batches of Lactobacillus reuteri can inhibit the response observed upon activating this pain receptor with its agonist capsaicin. This method is interesting because TRPV1 has, for instance, been associated with chronic and neuropathic pain and is expressed in a large variety of tissues and organs. In addition to these cell models, we also investigate tolerance to bile and acid, a topic which I have addressed earlier and will come back to shortly. Following observations in these models that imply increased bioactivity, we will try to isolate these probiotic effector molecules, conferring the effect. Although we are yet to find that increased bioactivity, and thus proteomics are not part of this current workflow, but it surely will be part of it in the upcoming years. We've observed that there is clearly an impact on bioactivity when we alter the means of production, which is visualized in this graph, where 3 differently produced batches of Lactobacillus reuteri DSM 17938 have been exposed to 1% porcine bile for a total time of 4 hours. What we can see is that batch 1 has an average 20% better survival than the other batches and than freshly cultivated bacteria. Now we're looking into how and why there is such an increase in survival, but presently we cannot explain the reason. One could speculate that this batch is in general more tolerant than the others, which is a question that we've started to address by testing the very same batches in the acid tolerance assay. However, it seems like the effect is limited to bile in this case, as batch number 1 is not surviving any better in the acid than the other batches. However, we intend to try these batches further in our models. I would also like to address a question that I've realized can arise with this method. Namely, why are we using porcine bile instead of the more commonly practiced bovine? The short answer to that question is that porcine bile is more potent than bovine bile, and we want to expose our bacteria to a harsh environment to truly test their tolerance, and that is why we use porcine bile. On the next slide, you will see my poster in its entirety, and if you are interested in a certain part of my presentation or have any questions, please don't hesitate to email me. Thank you very much for listening.